Okay, class, now where we're on is chemical digestion and absorption of lipids. The first thing you do, if you recall, pre-treatment, and that's emulsification by the bile salts. Emulsification by the bile salts. Remember, in order to get it underneath the water layer. That was the first thing. Then you had enzymes. You had some salivary amylase. We bring it up pancreatic, but you had salivary amylase. Didn't do a lot. If you're dealing with the triglycerides, remember the triglycerides have 3 fatty acids and glycerol. 3 fatty acids and glycerol. So what you have to do is take the 3 fatty acids off the glycerol. Triglyceride. Take the 3 fatty acids, and then you absorb that into the capillary villi. And you can read on that. Okay. So absorption of monoglycerides and fatty acids. What happens is that in the enzymes take 2 fatty acids off of the glycerol to make a monoglyceride, and then 2 fatty acids. They cluster with bile salts and lecithin to form micelles, release micelles to diffuse into the epithelial tissue, and combine with proteins to form chylomicrons. Now, my best description of this is in my serum lipid transfer. So what we had was, in my complete story, if you really want to know the complete story, you can come here to my serum lipid transport, and here would be eat meal with cholesterol, triglycerides, fatty acids. Triglycerides are broken down to a monoglyceride and 2 fatty acids by pancreatic lipase, some salivary also. All of the lipids stay as they are. So in other words, the only ones that you're really going to break down are really the triglycerides. Now, a triglyceride, remember, looks like this. It's glycerol with 3 fatty acids on it. 3 fatty acids. So what you do, you take 2 of the fatty acids off, and that leaves a monoglyceride. And that's the way it's absorbed into here. Okay? Fatty acids and monoglycerides leave the micelles. The micelles, so we go back to my complete story, in order to absorb lipids, need bile to what? Emulsify the lipids so as to allow them to pass below the water layer line in the intestine. Bile enters the first part of the intestines through that hepatopancreatic duct, the duodenum from the hepatopancreatic duct. Bile mixes with the lipids, forming these micelles. The micelles allow the fat to move to the top of the intestinal epithelial cells. See that? Okay, so we go a little further. All right. The lipids are allowed into the intestinal epithelial cells by simple diffusion, because remember, lipids can pass through cells very easily, particularly in the ileum, last part. The bile forming the micelle will stay behind, and some is absorbed later in the small intestine and large intestine. That's what we call the intrahepatic circulation. And some is eliminated in the feces. Remember, the large intestine, normal florobacteria convert some of the primary acids, primary bile acids, into secondary acids, and half of the bilirubin was converted to urobilinogen. Once the fats are in the intestinal epithelial cells, the monoglycerides are reunited to form triglycerides, and all the lipids are packaged in this we call a chylomicron. That is your first lipoprotein. Remember, we've talked about this. That's your first lipoprotein, the chylomicron. Okay? The chylomicrons are too large to diffuse through the intestinal epithelial cell, so they go out through the back of it. Here's the, right here. They're too large. to go. So they go out through the basement membrane by exocytosis. The chylomicrons then enter the central lacteal, right there, of the lymphatics, and it's carried to the bloodstream in the thoracic duct. Okay, so that's what basically happens. So let's go back to this one. So that gives you a more thorough explanation of this. And that thorough explanation is in my serum lipid transport PowerPoint. All right. So here we are. Okay, that's that. Passage of short chains is quite different from what we've described. These fat breakdowns do not depend on the presence of micelles, or not recombined. These short chain fatty acids, little small ones. All right, where are we now? Enzymes. Okay, now, so that was lipids. So we got lipids into the circulation through the thoracic duct. And then what happens to the lipids? They would enter cells and then go through the citric acid cycle we talked about and so forth. Okay, because remember, everything can go through, when we did this, fats also can go through, the glycerol can go into the glycolysis, and the fats can enter later somewhere in the Krebs cycle. Okay? So that's lipids. And like I say, my serum lipid transport PowerPoint is excellent to look at. All right, chemical digestion and absorption of nucleic acid. Enzyme, nucleic acid, RNA and DNA. So we have a pancreatic ribonuclease and a deoxyribonuclease break into nucleotides. Remember, a nucleotide is made up. What did we say? Let's go back here. Nucleotides, a sugar, a phosphate group, and a nitrogenous base. The nucleotide is the monomer. So pancreatic break into nucleotides. Intestinal brush border enzymes, nucleodases, and phosphatases. Okay, the nucleodases will break the what? Will break the sugar from the nitrogenous base, and the phosphatase will break the phosphate off. So in other words, it's going to split into three pieces. You see, a nucleoside, let me give you a term. A nucleoside is a nitrogenous base hooked to the sugar. When you hook the phosphate group, it becomes a nucleotide. It becomes a nucleotide. Okay. Absorption, active transport. Transported to the liver via the portal vein because it's water soluble. Okay. Those are nitrogenous bases. And so you can look at this. Vitamin absorption in the small intestine. Fat-soluble vitamins A, D, and K are carried by micelles because, see, they're fat-soluble. Water-soluble are absorbed, and they're going to go into what? The bloodstream, right? Because they're water-soluble. B12 has to bind with intrinsic factor. Everybody remember, it has to bind with intrinsic factor produced by the parietal cells and absorbed by endocytosis in the terminal ileum. But you have to have what? You have to have intrinsic factor produced by the parietal cells. In large intestine with vitamin K and B. Vitamin K is made by bacteria in the large intestine and some B vitamins also. And then they have to be absorbed. Electrolyte, mostly along the length of the small intestine. Iron and calcium absorbed mainly in the duodenum. Okay. Sodium is coupled with absorption of glucose and amino acids. So this kind of gives you how you get the electrolytes in. See, that opens up a story because calcium, remember, parathyroid hormone works with calcium. Okay. For iron, we have ferroportin, and different things. So it gets a little more detailed, but again, we're looking at the overview. Ionic iron is stored in mucosal cells with ferritin. We talked about that when we did the iron. Vitamin D and PTH. Okay, so really this ties in. If you go back and you look at your other. Okay, we already talked about sodium potassium pump. That's what sets up the gradient for all of these things to do secondary transport. Iron absorption is brought in cell through transport involving a transporter, DMT1. Iron absorption, ferritin, ferroportin may leave the end and enter the body via basal ladder known as ferroportin. So we won't talk a whole lot about iron. We talked about it before. If you want to talk about that, we can do that at another time. Water absorption, 95% absorbed in the small intestine by osmosis. There's some taken up in the large intestine, remember. Okay, that's water absorption. Malabsorption, anything that interferes with the delivery of bile or pancreatic juice. Because if you don't get the bile, you can't get the lipids. If you don't have the pancreatic juice, you can't do the others because you can't break it down to the monomer. Okay. Also, you could have trouble with damaged intestinal mucosa that could cause a problem. Gluten sensitive, you heard gluten sensitivity. Okay, celiac disease. Gluten is a substance that helps bread to stick together and so forth. Gluten damage intestinal and brush border. It's more of an autoimmune thing. Grains and rice. Okay, we won't get into the developmental. So we've talked about, and you can kind of glance at that, 95 in the small intestine, but then the rest in the large. You leave about, we said, 150 cc's with your feces. 150 cc's with your feces. Okay. And again, this is iron. Inflammation causes iron to not be released from cells as much. Inflammation. And that would get into something called hepcidin and ferroportin, which I'm not going to get into. That gets real detailed. But the reason in inflammation iron is held in the cells and not let out in the bloodstream that much is because bacteria like to use iron. So you try to starve them of their iron. So that gets into an interesting discussion. Okay, we're going to close here for the gastrointestinal system.